Hi everybody. We're going to continue on with the DNA replication lecture that we started last time. I wanted to also say that I'm going to post a video on Canvas with the links that I give you, a video of DNA replication with the links that I give you of this lecture. And then your test will be on Wednesday, and I will send you more of an update about that test. You will need for this lecture, you will need the handout that I handed to you last time about CAM plants and C4 plants. And on the back is a list of all of the enzymes and molecules that are part of the DNA replication. You might remember from last time that we were talking about how DNA was built and the fact that adenine and thymine always join and guanine and cytosine always join because of the number of hydrogen bonds between them. Also remember that there were different ways that the sugar's backbone went, that is that they are anti-parallel. One side where the sugar backbone is going in the 5-3 direction and the other side where it's going in the 3-5 direction. So I'll tell you more about why that is important pretty soon. Just a reminder, this is called semi-conservative replication. It has to do with what you see up on the whiteboard right now. This is talking about how the old strand was in the blue and the new strand is in the orange. What that means is that half of the original DNA molecule is kept intact while the new DNA molecule is attached. And when I get all done, I'm going to have two separate strands. They will be attached at a centromere, and we will talk more about their structure when we talk about mitosis. Right, I'm going to change out the overhead. This overhead, what this is depicting is it's showing you how the sugar phosphates become attached to each other. So basically it's talking about how the removal, or that is the phosphate from ATP, is going to be the phosphate that is part of the sugar. So just interesting. Another thing to know is that the DNA molecule is going to be copied, unlike what we talked about in class last time when we talked about it unzipped and it got copied all the way up just like a zipper. That was a simplification. What actually happens is it starts in various places along the DNA molecule, say for example 100 places. And each of those is called a replication bubble. Each bubble will proceed in the replication and eventually meet up with the adjoining bubble. So this bubble, as it goes, will meet up with this one. And as we get all done, we're going to end up with two daughter molecules there. Okay? All right. So, first of all, let's just talk about how on the DNA, that has nothing to do with this, per se, so I'll just stand in front of it. The first thing that happens is on the DNA molecule, the bubble is started by something called an initiation protein. There are sequences on the DNA that will depict where the initiation protein will attach. And when that attaches, an enzyme called helicase will come in and undo the molecule. So over here on the board, I'll just move this so you guys can see that. Over here on the board is a list of the molecules in question. So we have the initiation protein, and then we have the helicase. So the helicase is going to undo the DNA molecule, and if it just undid it and moved along and undid some more further down the line, what would happen is the molecule would just go right back together with its hydrogen bonds. So it needs something to keep it apart, and those are the single-strand binding proteins. The single-strand binding proteins just keep the molecule separated, right? The next enzyme that's going to be necessary is called primase. So now we can go back to the overhead. Okay, so right here you can see there is on the DNA molecule, if this is where my replication is beginning, notice that this would be the 3-5 side of the original strand, and that the copying can only happen in the 5-3 direction. In other words, it can't really shake hands going the other direction. It can't add on to anything but the 3' end. It has to add on to the 3' end of the original molecule. So that means it has to go this way. If the molecule or the other side was, if we were trying to go in the same way, it would be the 5' end, and that would not work. All right, so we'll talk about that a little more later. The first thing that happens is that primase, an enzyme, is going to lay down a primer. Now that primer is a piece of RNA. So you might remember from our discussion last time that RNA is slightly different than DNA in 2 ways. One way is RNA does not have thymine. It has uracil instead. So if it was base pairing, which is what it's going to be doing here, for example, if that was adenine, instead of pairing with thymine, it would be pairing with uracil. And guanine and cytosine are still a pair, even in RNA. The other difference that RNA has is it is a single strand. It is not a double helix like the DNA molecule that we talked about last time. So to repeat, primase is going to lay down a primer. The primer is kind of like the beginning of a zipper. That is where DNA polymerase, which is the enzyme that is going to copy the actual DNA or the template DNA, that enzyme can only attach at the primer. It can't just come along and attach randomly any place. It has to have this beginning primer to go. So what's going to happen next then is you have the RNA primer, and then you're going to have the DNA polymerase is going to come in and move along and copy by base pairing all the way along in the 5-3 direction. Now this strand that can be copied in a continuous sweep because it's going in the 5-3 direction is called the leading strand. All right, so this is the leading strand. Okay, so that's how that's going to happen. Now, if another replication bubble came in here, what's going to happen? You know how we said that the 2 bubbles might meet. If the next bubble comes in, as the DNA polymerase comes through to the primer, it will just copy that piece of primer. Then what you're left with is 2 little pieces that are left over, and those will be joined by an enzyme called ligase. So ligase is like a glue. It's going to glue the 2 pieces together. All right, so that is the leading strand. Now we need to talk about the other side. Okay, so now that we are looking at this, notice that this is the original 3-5 piece, and this was the original 5-3 direction that we're going. This overhead would be a lot more complete, in my opinion, if it had a primer up here, so that you could see that that was the leading strand starting at the primer and continuously copying all the way down. So this part right here is the leading strand. Now the problem is that as I start copying, what was happening down here, because this is the 5-3 direction on the original strands, I cannot copy that direction. So what has to happen is that I have to lay down a primer and copy back. And then I'm going to lay down a primer and copy back. They kind of already took out the primers here because the DNA polymerase is replacing them. Right here is showing a primer, copying back, primer, copying back. These are called Okazaki fragments, and this is called the lagging strand. So on the lagging strand, you actually have to open the DNA molecule a little bit, and as the progression goes, this one has to start it and come back, and then it goes along further, start, come back, right? So this is just because of this copying direction has to be this way. So in a replication bubble, you are both going this way and this way in your copying process, all right? So just to reiterate, basically what has to happen is at the beginning, an initiation protein comes in. It identifies where we're going to start the replication bubble. Then the helicase comes in and breaks apart the DNA molecule. The third thing that happens is the single strand binding proteins will hold them apart. And then the primase lays down a primer, and DNA polymerase can come through and copy on the leading strand. If it's a lagging strand, it has to wait till it opens a little bit. The primer is then laid down a little ways and copies back with the DNA polymerase. Notice there's several DNA polymerases here. If we were getting more technical, we would talk about the names of those different ones. They actually have DNA polymerase 1, 2, and 3, etc. Not a detail we're going to cover. All right, now a couple of other interesting things. Should I stop? So a couple of interesting things is that if I were to pull a piece of rope and it was wound up, you know how rope is wound like this. So that's basically like a helix. If I were to just start pulling it apart, what happens is that it will start bunching up and it won't come apart very easily. And that is exactly what would happen to the DNA molecule if I were to just take helicase and go straight down the replication bubble all the way. It would start getting bunched up. So what I have to do is I have to cut the DNA and let it unwind various places along the way. This is another enzyme called topoisomerase. You might remember that an isomer is something that is, the mirror image of the molecule. In this case, this is just like saying that I can cut it and it'll unwind and reattach. So it's not like that's just the name of the enzyme. All right, so that's what that is doing. And then there is another enzyme called nuclease. Nuclease will come in and repair any problems with the replication process. So let's say, for example, I had a mismatched base pair right here. Nuclease travels along here, it identifies a mismatch, and will cut it out and replace it. So nuclease is like our repair enzyme, which is why really the replication process doesn't have many mistakes. It basically pretty much it cuts and repairs itself if there's a problem. One last thing that I want to talk about is something called a telomere.